Did you know that Atlantic salmon, a large female Atlantic salmon, can lay as many as 10,000 eggs? Extraordinary. And if you sort of put it down to a very simplistic perspective, essentially we just need 2 of these back to sustain our populations, if you really boil it down. So we just need 2 of 10,000 for the large ones, or maybe it's 2 or 3,000. It's still a very small proportion. We need to come back and successfully reproduce. We've seen numbers of these versions of these ICES, which are the best international estimates of the salmon population in the wild at the moment. And it's quite clear if we look for the last 30 years, we just haven't been getting 2 back. Extraordinary. So yes, I'm based on the River Frome in Dorset in the south of England, where we work. The River Frome is chalk stream. A different beast, if you like, to some of the rivers that you see up here. But whereas there are many things that are different, there are certainly also a number of similarities. My group has been working on the salmon population for, I suppose, over 50 years, and it's now nearly 50 years. We nearly have the 50th anniversary of our resistivity fish counter that estimates the number of returning adult salmon on the river. What I will say there is that that's very nice to have such a long time series, but when the crash happened in the Atlantic salmon population in the early 90s, in the southern part of its distribution at least, the group very visionarily thought, well, actually it's nice to have a measure of what the population is doing, but just measuring how many adults return doesn't really tell us anything about where and when the issues, the new issues, if you like, of these fish occur. So what we started in 2005, or was it 4? I forget now. We started utilizing pit tags. There's been some talk about them earlier. And what we do, or have done annually since then, is we pit tag 10,000 young of the year salmon on the River Frome. And I should just put that into perspective here, that the River Frome is a very productive system. It sits at towards the southern limit of Atlantic salmon's distribution. So our fish grow very fast, and over 97% of our salmon leave the river after just 12 months in the river, so they grow fast. Why do we like these pit tags? Well, we've seen previously there are individual identifiers. We know Bob from Amanda, and we know where and when they're tagged. And when we tag them, we take lots of biometric measures, we take scale samples, etc. So we know a lot about these individuals. But what they also do is, what they provide us with, in addition to the data we get from our resistivity counter, is we get population estimates at different times of the life cycle. So that's at the par stage, the smolt stage, and we even get a second estimate of the adult returns from these pit tags. Because in principle, these tags stay with the fish for life once they're tagged. But what they also do, which is really important, and I think is one of the areas where we can start to take things a bit further, is that they provide life history of individuals. So we can start looking at what happened to Bob and what happened to Amanda, and think about what was different in different parts of the river, or between different years, and why, how does that affect things? Right. So what I'm going to do today is I'm sort of going to do a summing up of some of the things we've learned over the last 3 or 4 years, really, where we've been starting to sort of pull this data together for understand on looking at what affects numbers and what affects the quality of these fish, if you like. So that's what I'm going to go through here. Now, the River Frome is a very different system to your rivers in the northeast of Iceland, as I said, but there are still things that we can learn here. Okay, one of the things that we get from this exercise, and as Peter said, I've just spent the last 3 weeks on the river, so if I've got a bit of a tan, it's a farmer's tan. I can assure you there's no tan below above the elbows. Anyway, we get an estimate of how many young of the year salmon are in the river. And on the River Frome in September, when we go out and do this tagging exercise, generally speaking, we have about 100,000 young salmon in the river at that time of year. But as you see from the top graph here, there is some variation. And particularly what you'll notice in 2016, we saw very low numbers. And that was in fact something that was reported throughout the region, shall we call it, England, Wales, Scotland, Ireland, France. Something happened that year. So that is obviously of interest. But what we did this year, actually, we then tried and pull some of the data together and have a look at least at the things that we do measure in the river, things like river temperature, discharge, and how many fish are there. Which ones of these parameters are actually important for the recruitment? So how many fish do we have from the eggs that are laid in the winter? How many fish do we actually have from that come the next autumn? And the three plots below, I'll point your attention especially to the one warm spring and cold emergence temperatures. And from the data, it was very, very clear that for our fish on the River Frome, the temperature around spawning and again at emergence time were really crucial. Warm temperatures around spawning was very bad, as was cold temperatures around the time when these fish come up and start feeding. The middle graph at the bottom there, you'll see we also had a look and see what effects are there of flood events. There's a lot of talk about egg washout and so on. The winter 15-16 was very wet, so we had lots of water in the river, but on the River Frome there was really not an effect of these floods. And that's maybe not so strange given the fact that it's a low gradient, lowland river, so it just doesn't have the power that these sort of higher gradient rivers has. But more surprisingly is the graph on the right, where we have an estimate of the number of eggs laid from the number of adults that returned to the river the previous year and the size distribution, etc. of those. And what we see here is that there was actually no relationship between the number of eggs laid and the number of fish, the young fish that were present the next year. Now that was at first glance definitely took me by surprise. I hadn't expected that. But I think what we need to bear in mind here is that we have a couple of unusual years, climatically unusual years, winter 15-16 and again in 20 actually. So it's quite clear that we don't see a relationship, and I hazard say that it's simply because some of these climatic conditions override any effects that we might have that we're expecting from density and so on. They can simply be more important, these density independent effects. We were also contracted by Natural Resources Wales, which is essentially the sort of the body in Wales that looks at the natural resource, so their environment essentially. And we did very much the same exercise here for I think 7 of their main salmon rivers. Now there's a slightly longer time scale for this. It's about 2 decades worth of data, but quite reassuringly we see the same effect of emergence temperature and of spawning temperature. So our too high spawning temperatures are not good for these fish, and neither are low emergence temperatures. And I think here we need to bear in mind that what's important, I should really sort of emphasize here, is that the River Frome is a chalk stream. Chalk streams are primarily groundwater fed, so the majority of the water running in the river has simply come out from the springs. So we have very stable temperature regimes and we have very stable flow regimes, whereas the Welsh rivers are much more dominated by surface runoff, so rainwater essentially. So much higher fluctuations in their temperature regimes as well as in the discharge in the rivers. So they're very different types of rivers, but we're still seeing some of the same main effects. And what we see here for the egg deposition for these rivers, actually, we do see what we would sort of expect from the Ricker curves that was presented earlier. So that sort of illustrates these density-dependent effects are important, but in some years it'll just be overwritten by climatic changes that really is not related to the density of eggs or density of fish as such. Now, that was a bit about how many fish we get, what's the recruitment like, what might affect that in a system like ours anyway, or in the region that we work in. Now, what we've learned lately is that, it's not just a numbers game. It's not just about getting as many par or as many smolts as you like. And how we learned that? Well, I'll just remind you that these pit tags are unique identifiers, so we know the individuals from each other. And what we do every spring is that we divert our salmon smolts into a carrier of the river where we trap a proportion of them. So we know what size Bob and Amanda were when they were caught on their way to sea, and then we've been able to then look at what is their probability of returning to the river. Luna spoke about this earlier as an established fact, and it's interesting that it's been a hypothesis that's been out there for a long time. Bigger smolts must be better. But actually, we were among the first to show with this time series of data we have, where we know the individuals, that yeah, indeed it is the case. And what we see here on this graph on the x-axis is the size of the smolt, and the 1-year-old smolts in the River Froome, which is by far the dominating age group, they sort of range naturally between 12 and 16 centimeters. And I'll just point your attention to the y-axis, which is the proportion of them essentially that returns. And in fact, there's more than a 3-fold increase in the probability of returning if you're towards the upper end of the size spectrum compared to if you're at the lower end. So it is not just about numbers, it's also about the quality of these smolts. Right. Let's go a few years back where we worked and published some work on the size of our par in the river. And what you see here is a time series of how big are they when we go out and tag them. But you'll see a couple of additional rivers here, the River Loire and Scorff, which are in northwest France, where they've also monitored their salmon populations for a number of years. And this data was from an EU Interreg project called Morefish. And what we looked at there was what sort of effects, what size these fish are towards the end of their first summer. Now, these are, again, they are all young of the year fish, so these are 6-month-old fish, all of these. And the take-home message from this slide really is, in the last sort of 20 years or so, the trend has been the same in this region. The par are getting smaller. So par of the same age, they're not growing as fast as they used to. Now, of course, we're interested in knowing why is that and what's affecting it. So here's sort of some of the core results from that paper in terms of that. And if you just pay attention to the solid lines and don't pay so much attention to the dotted lines, what you see essentially on the left there is that density is important. So all 3 rivers, there were some between river differences, but in principle, all 3 rivers showed that the size of these fish is to some degree dictated by the density of fish there. But one of the other interesting things that was pulled out was that the summer mean discharge, minimum discharge, was also important. So effectively what you're looking at there in that middle graph is that the higher the minimum flow we had during the summer, the bigger these fish were. So in years where we didn't have very much water in the river, the fish just didn't grow as well. There probably wasn't as many resources available, and there definitely wasn't as much habitat available. So that's important. And the third one is also very interesting, and Luna talked about that for some of the Icelandic rivers, and here's an example of the same being important, winter degree days in terms of essentially the cumulative number of degrees temperature over the winter is important. And you've got to remember these fish that we're looking at here, they're young of the year. The only winter they've experienced was the winter when they were either eggs or yolk sac elements in the gravel. So, like Luna, earlier in the year these fish come out, the bigger they grow by the end of the summer. So it's important. Right. Here's data from one of our current PhD students who's working on a big project international project that we're working on at the moment. And this is a graph from a paper where she's looked at the phenology and the timing of the smolts leaving the River Froome. So this is all data based on the pit tags when they're detected leaving the river. And the take-home message from this is that you'll see over the last sort of 15 years, our fish have gotten, 7-8 centimeters smaller on average, our smolts. So that's worrying when you think about the slide I presented earlier showing that the size of these fish when they leave the river is important in terms of their probability of returning to the river. It's quite worrying, really. And sort of if we dare speculate a bit on why could that be? I mean, so obviously what this paper also shows, the paper also shows is that it's the cumulative temperature in late winter and early spring. That's what dictates at what time these fish leave, in our system at least. So essentially what we've seen over the last couple of decades is that our late winter, early spring has become warmer, which has meant that the fish have been ready to go earlier. Now, why is that interesting? Why is it interesting that they're going earlier? Well, Stephen McCormack, I think, framed the phrase smolt window in the late 90s, and here's a little illustration from that paper that he published, which has since been quoted loads. And on the left-hand side, you sort of see on the top one is a graph of how good are the environmental conditions during the different weeks of the smolt run. And we're looking at the environmental conditions here in the early sea area, so where they go after they've left the river. So this is sort of illustrative, really. And the bottom one is the timing of when the salmon smolts leave. And when the one on the right then shows you the effect of if you shift either one of those to a couple of weeks, you see a very, very significant reduction in the survival of these fish, so in the return rate of them back to the river. So this is where us seeing that the river dictates that they're ready to leave earlier. What we essentially need to look at there is there then a mismatch in the timing of when are the optimal conditions at sea. And we don't really have the answer to that. But actually, I'll go on now to talk about another PhD student who's based in France, but another part is part of the Interreg project, Cécile Trehine, and she's published a very, very nice paper last year. It was this year, actually, it came out. And what she's done is that she's utilized the fact that scales tells us a bit about how they tell us about how fish grow, and they're like the trunk of a tree. You can cut it and see when winter how old it is, because winter growth is slower in the winter. And the scale you see here is taken from a returning salmon, a 2C winter salmon, so a salmon that spent a couple of years at sea. And what Cécile did was that she took advantage of the fact that the sort of diameter increase in the size of this scale is directly related to the size of their body. So she was back calculating essentially what the size was at a given stage from the growth on the scale. And she did this work. She's including all the 5 rivers that we work on in the English Channel area, but the particular work that this publication is on the River Saloon in France. So she looked at growth at different stages while these fish are at sea. And before we go on to the graph, I should say that she didn't find any difference over the sort of couple of decades that she looked at. Now, it's important to say that there's a long tradition of collecting scales. So she had scales going back, I think, about 30 years or so she could look at how this marine growth had been changing over time. And where she found the big differences lately is that over the sort of last 10, 15 years, as you see in this graph here, there's been quite a dramatic decrease in the growth of Atlantic salmon from the River Saloon in the first 6 months while they're at sea. So the first summer at sea, they have really, they're not growing as fast on average as they used to be. And you'll see here that this signal is pretty much the same for the fish that stay out 1 year and 2 years. That first year, they're just not growing as well as they used to, at least in some years. And why is this important? Well, if the fish return smaller to the river, then of course that means, particularly for the female, they're less fecund because the number of eggs they have are directly related to their body size. The smaller returning fish are going to mean less reproductive potential for the females in particular. But what she also found, and which is interesting, and it's about 8 years ago now that we finally got a technique where we could non-destructively sex these fish using DNA. Previously, we had to literally cut them open to look at their gonads to see whether it was a male or female, at least for the juveniles. But so Cécile took advantage of this because the scales still, even 20-year-old scales, still have DNA on them. So she was able to sex all these individuals that she'd measured the growth rate on. And the second graph here looks at what does it take for, how much growth does it take for an individual to return to the river after only 1 winter at sea? And the first take-home message here is that, yeah, the more they grow, the more likely they will. So on the y-axis, the probability of returning after just 1 year. The faster their growth during that first summer at sea, the more likely they are to return. But this is sex specific. So essentially, the females need to grow faster that first year to make that decision to return. There are no doubt a genetic element in terms of whether they're predisposed to return as 1 or 2 years old too, but there is clearly here also a sort of an ecological element. And what's the knock-on effect of them getting small or growing less, shall we say, in that first year is that they will have implications for the sort of population dynamics. At what age do they return? And why is that important? Well, if they spend another year at sea, they got another year to die out there essentially. But the flip side of the coin is that if they spend another year out there, they will be bigger when they return, the females, and therefore they'll be more fecund. So it's not necessarily a straight relationship, but at least this is really important information in terms of managing these populations going forward. And particularly if we can start linking what is it that makes them grow fast. If we already, during that first summer, can start measuring somehow something out there that tells us about how fast our fish probably going out there now, then we'll already be able to sort of account for that effect of when and how big will they be when they return in terms of our management. That's quite interesting. Now, I talked a lot about the sort of density independent, the climate driven things, but I just want to sort of the final slides here talk about a PhD student of mine who's just finished and who's done some really nice work. Here you'll see a picture of the River Frome. It's a chalk stream, and this is what it looks like in a nice piece of habitat on the River Frome. Very clear water, lots of plants. And it's interesting, Ian asked a question earlier today about the sort of complexity of the habitat. And what I should explain is because the River Frome is a low energy system, the largest natural substrate in there is flint about the size of a fist. So we just don't have big boulders there. It can become, if we don't have the plants that we have in the river, they create the heterogeneity in both habitat, but also in flow regime. And also it creates this three-dimensional habitat that I think impacts the behavior of the salmon more, because they are very territorial at that sort of summer stage when they have their habitats. So what we'd observed over the first years of our tagging program was actually, if we turned up at a site where there wasn't much ranunculus there, well, we probably weren't going to catch very many salmon. But if we turned up at a site where there was a lot of ranunculus, well, then at least there was a probability that we might catch a lot there. There might be a high density. So Jessica got the PhD to look at this, where I'd got the funds to look at this, and yes, she looked at about 25 sites across our catchment, and as you see on the top left graph, there was a relationship between the amount of ranunculus, a positive relationship between the amount of ranunculus and the density of young of the year salmon. There were a number of other factors that also came through in the model selection exercise, and there was things such as distance from the tidal limit, and very naturally the distance from the nearest upstream salmon redds. Now, when you just go out and measure a parameter here and relate it becomes a correlation. So they were related to this ranunculus, but there could have been so many other things. So what we did to try and take it a step further towards cause and effect, so away from the correlation, was that we established some experiments where for a number of sections, we removed the majority of the ranunculus and planted that in others we left the ranunculus in the solid density it was. So we had effectively 3 different treatments of ranunculus. And here again, we're looking at the number of juveniles that we found, and now we're looking at them in these experimental sections. And what we found was that in the summer, there was a very strong positive relationship between the amount of ranunculus and the density of juvenile salmon that we had there. But what we also found was that when we got to the autumn, when the plants start to die back and the salmon start to look for maybe slightly different habitat as the water cools down and the metabolism slows down, was that we didn't find an effect later on. So this habitat was important for them during their summer feeding in this highly density dependent period of their life. But another very interesting finding that we did in that experiment, because all these fish at first capture were tagged, so we were able to identify the different individuals, and what we found was the fidelity to a site was much higher if there was related to the amount of ranunculus. So essentially, if we had a lot of ranunculus there, so a lot of good structure in the habitat, then the fish were more likely to stay in the same patch, if you like. But more importantly was that the growth rate was also related. So the growth rate of our salmon, not so much the trout, but the growth rate of our salmon was also very much positively related to the density of the ranunculus. So in effect, what this additional complexity in the habitat managed to achieve was that we were able to have a higher number of fish there, but not only a higher number, we also had faster growing fish there. So takes it away from a numbers game a bit. Now, I'm not advocating that the ranunculus should be planted elsewhere, but it just shows what potentially can be done at this density-dependent stage of their life. Another interesting finding that I'll just sort of finish on that experiment was that adding this additional structure to the habitat was that the fish were feeding better. We found that the salmon, we did gut flushing of these individuals, and they were feeding better. They got more food, which links clearly to the fact that they were growing faster. But what we also found was the niche overlap in terms of what they were eating with the trout was reduced when we had more complexity in the habitat. So effectively, we weren't just benefiting the individual species, but the fact that it sort of potentially removed some restrictions on feeding that were coming from other competitors in the system was also relaxed. So this competition was relaxed by adding this complexity. Okay, so sort of concluding on what we have experienced in our system is that recruitment, yes, we have some very important climatic related variables that clearly to some degree control what the productivity of our system is. But that's not everything. And you know, as soon as we mention the word climate, I think a lot of people sort of think that, okay, that's not anything, we can't do anything about that. But I mean, I'll just refer to Nikolai's earlier talk, and there might clearly be some measures we can take to try and ameliorate some of these effects of extreme weather. But maybe it is easier to look at some of these biological variables and try and help them at this density-dependent part of their life. And clearly, it's very important, as Jessica's experiment illustrated, that we must see this through an ecosystem lens. It's not enough to just look at these salmon as in isolation. And I think that's come through a lot of the talks today, that we must really look at them as part of being part of an ecosystem that needs to work. And the other thing that we've illustrated by the fact that you saw clearly with the relationship between the size of the smolts and their probability of returning from sea is that we mustn't see any of these stages in isolation. It's a part of a life cycle, and there will be legacy effects. Now, earlier today, there's been talk about bigger smolts probably being able to better avoid predators. That's very true. But I think also we must also think about it as them bringing a bigger lunch pack. So should they meet a dry patch, if you like, a patch in their early marine stage where there just isn't very much food available, well, then at least they have a bigger lunch pack to maybe get through that and get back again. So, yeah. And my very last slide, I'll just go back to this one because, yeah, I didn't have a stock image of a very large female salmon, so I Googled large female salmon, so that was like, oh my God. Okay, I'm not even sure this is a female salmon, but it's a nice example. But the reason for returning to that slide really is, you know, there is this enormous reproductive potential in this species. So if we can get at least some elements of their life cycle right, then they have this potential to bounce back. And we see that in places where the environment has been cleaned up. My home country, Denmark, is a prime example of some rivers have been made more accessible to the salmon and the water quality better, and the salmon are now thriving there. So there is this potential for them to bounce back. We mustn't forget that. Thank you.
Thanks very much, Rasmus.
Thanks.